Lecture 5 will be an examination of the structure of the phospholipid bilayer membranes that make up both the interior endomembrane system and the plasma membrane that surrounds all cells. If you're taking this course over the summer, this represents the end of lecture material for Exam 1. And tomorrow, Exam 1 will open up for you to be able to take on Web Campus. There will also be an additional quiz that's available on Socrative. In replacement of doing an actual recitation material, this is an opportunity for you to test yourself with all the material that's led up to this point in the course, and it should be done before you try to take that online exam. So let's start by going through some review questions. There are 5 questions, so please log into Socrative and pause the video here for as long as you need. So let's discuss which organelle produces energy in the form of ATP that's necessary for the cell. That would be mitochondria. And in order for us to better understand how mitochondria produces ATP, which is going to be a big component for the next section of this course, we need to understand their structure. When we look at the structures of the mitochondria, recognize that while many of these structures look very similar to a chloroplast, which is also an endosymbiotic organelle, there are no thylakoids. Thylakoids are the structures that form the internal membranes of these photosynthetic organelles. But mitochondria fold their inner membrane into structures known as cristae. These look different, they're arranged different, and they have different proteins in them than a thylakoid would. What's the goo that fills the mitochondria? Well, once this was known as a cytoplasm, because mitochondria used to be individual cells. But when those cells became engulfed and they can no longer reproduce on their own, we can't call this a cytoplasm because cyto refers to cell. And this is not actually going to be material of a cell. This is material within an organelle. So we've renamed that goo the mitochondrial matrix. Which cytoskeletal component supports cell shape? It would be all of them. But it's also important to recognize that while all cytoskeletal components support cell shape, it's the defining factor that causes us to include them in that category, that classification, that each cytoskeletal component has its own unique functions that it carries out in addition to simply providing structure. This would include how microtubules are going to be the train tracks along which vesicles move, as well as the spindle fibers that divide chromosomes during cell division. We have microfilaments, which form the cortex of the cell, capable of allowing the cell to produce pseudopodia as a form of movement, or to contract in a muscular sense. And then there are the intermediate filaments, which are more stable forms. They're not as dynamic as microtubules and microfilaments, but they form that nuclear lamina, which is so critical for nuclear shape and support. Cells that lack a covering, that elaborate network, which we call the extracellular matrix, are still able to survive despite the fact that they do not have a cell wall. Extracellular matrix does not substitute a cell wall, but it provides many of the functions to even an unwalled cell, allowing it to interact with its environment and connect with other neighboring cells. So our goals for you today will be to apply that macromolecular descriptions that you know in these molecules to the composition of a phospholipid bilayer, and then explain how this allows the membrane to achieve its biological functions. Explain the fluid property of membranes and their importance to cellular functions. Describe the cellular environment inside and outside of the plasma membrane. List and describe functions of proteins embedded in the plasma membrane. Describe the methods used by molecules that cross the membrane, and to name osmotic environments and describe the effects of these environments on the cell. So we're actually going to start by taking a look at number 2 and recognizing the importance of membranes. Because that endomembrane system that we spent so much time focusing on in our last lecture really connects all of the internal components of the cell with that plasma membrane. When we take a look at this membrane, it's important to understand that membranes are fluids. They're actually about the consistency of olive oil. The reason that these membranes stay together, forming this layer that we know them as, is because that internal core of the membrane is hydrophobic and will not interact with the watery environments that are both inside and outside of the cell. But just like how if you take olive oil and try to mix it with water, it refuses to mix. If you try to mix olive oil with, let's say, liquid butter, you get an appropriate mixing. Membranes are capable of mixing together. So when you see this little green membrane barrier of this particular vesicle merging with the phospholipid bilayer of the plasma membrane, recognize that these are truly mixing. The proteins that were embedded into the membrane of the plasma membrane, the phospholipids that are there as well, are mixing and creating a new singular membrane that contains the components of both. You can see here how dynamic these interactions are. It's the hydrophobic regions at the core of this membrane where these proteins are being embedded. The fact that water does not readily pass through the membrane because it's not willing to interact with those hydrophobic tails at the core, but that the proteins provide channels for which these water molecules can move and span across the membranes. These are core concepts for us today. So let's take a look at the actual composition of this phospholipid bilayer. Recognize that the hydrophobic tails are interacting with each other. Since both of these regions are incapable of hydrogen bonding with water molecules, they are capable of interacting with each other. That's why they're called hydrophobic. The hydrophilic heads, on the other hand, can hydrogen bond with water. So they're going to prefer to interact with the watery environments either inside or outside of this membrane. The fact that we have both of these regions, both hydrophilic and hydrophobic regions, in the same molecule, we refer to these as amphipathic, meaning they're molecules with both properties. This phospholipid bilayer, while primarily composed of phospholipids, does not achieve all of the things that it does by just existing of phospholipids. I want to briefly introduce you to the importance of cholesterols, which are going to be hydrophobic macromolecules. These are lipid molecules that can embed themselves into a phospholipid bilayer. They can interact with hydrophobic tails, and their interactions help to properly space these phospholipids, allowing the membrane to be more liquid. It gives it some fluidity, but by having the proper levels of these cholesterols, it also helps to support the structure. Too many of them, it becomes too fluid. Too few, and it becomes too solid. But proteins can also interact with these phospholipid bilayers. Hydrophilic proteins might interact primarily with these heads, but you can also have hydrophobic membranes or, hydrophobic proteins, or amphipathic proteins that are capable of spanning the path of these membranes. We call these integral proteins because they actually integrate into the network of these membranes. And they're typically amphipathic in the same way that phospholipids are, capable of interacting with both hydrophilic regions at the outside of the membrane and hydrophobic regions on the inside. When we take a look at these proteins are going to have roles, but most of these proteins, or at least many of them, will be glycosylated because the way in which the proteins reach the plasma membrane is having been produced on the rough ER and inserted into the lumen of the rough endoplasmic reticulum, and then trafficked through the endomembrane system. There are also lipids that can be produced, and when they go through the smooth ER and the rest of this endoplasmic or endomembrane system, They can also have carbohydrates attached to them, and so these are known as glycolipids. These carbohydrates, which are attached to glycoproteins and glycolipids, are really important. They're like little flags on the surfaces of these cells, because what's luminal to the endomembrane system becomes extracellular after these membranes have fused. So cells can recognize each other by looking at these carbohydrates. Here's a real-world example. Carbohydrates are what determine your blood type. When you take a look at red blood cells, the carbohydrates that are being placed onto these protein and lipid molecules can either be A, B, both, or neither. If you have only type A carbohydrates, then you would be a type A blood. If you have only type B carbohydrates, you would be type B. If you have both of them, A and B, then that would allow your cell to be an AB blood group. And if you have neither, it doesn't mean you have no carbohydrates, but if you have neither A or B, that's known as type O. The reason why type O is a universal blood donor is because without either of these carbohydrates on the surface of their red blood cells, their red blood cells, can't be recognized as foreign. The reason why AB can only be donated to other ABs is that both A and B are present. So someone who's type O sees foreign, someone who's type A sees B and sees foreign, and someone who's type B sees A and sees foreign. So now I want you to think to yourself for a moment. The glycoprotein collagen is a critical component of the extracellular matrix on the outside of your cells. Would it be produced by free ribosomes or bound ribosomes? Well, if your cell is going to have proteins like collagen on the outside of the cell, then it's important for us to recognize that this cell has to be able to get collagen on the outside. Collagen gets on the outside because it was inside of a vesicle that merged with the plasma membrane and released its contents outside. In order for this protein to be produced inside of a vesicle, it needs to be produced in the lumen of the ER by a ribosome, which is attached to the surface of the ER. That means that collagen was produced by bound ribosomes. Now I want you to log into Socrative and answer this question for me. Pause here if you need more time. How about actin? Which ribosome population would produce actin, the monomer that's responsible for building microfilaments? Well, here's a more extensive view of that process. Here are proteins which are being produced in the lumen of the ER, being trafficked inside of vesicles, inside the lumen of the Golgi, inside the vesicles that bring it to the plasma membrane. And you can see here that these proteins inside the membrane, the interior region, actually becomes extracellular. Well, actin is going to be the monomer for microfilaments. And since microfilaments are going to be the network which is inside of the cell, then these need to be produced by free ribosomes. There are faces to membranes, insides and outsides. They don't just flip around. Things that are cytoplasmic stay cytoplasmic, and things that are luminal stay luminal. But if they traffic their way all the way to the plasma membrane, things that are luminal end up extracellular. This sidedness is very important because even though membranes mix horizontally, when you have this merger of membranes, such as a vesicle with the plasma membrane, they rarely flip to the other side. Things that are placed in a particular orientation stay in that orientation, and the result is that the components of a membrane are asymmetrically distributed based on the side in which they were first created. So true or false, glycoproteins can be found in the cytoplasm of the cell. Well, all carbohydrates are added in the lumen of the ER or the Golgi, which means that glycoproteins have to be produced in the lumen of the ER. That means that this is false, because glycoproteins are not being produced by these free ribosomes. These are going to be produced by bound ribosomes. If this isn't something that you understand, if this wasn't very easy for you to answer, you need to go back and review the endomembrane system. When we take a look at these membranes, I want you to recognize that membranes have functions, but that the functions are based on the components associated with the membrane and embedded into the membrane. We're going to focus on how proteins provide functions, because there are a lot of different functions for proteins, in part because the fact that we have 20 different amino acids which compose those proteins, we have many, many different properties which allow us to achieve many different shapes, and many shapes mean different functions. Peripheral proteins are going to be associated with the periphery, or the outside. They're bound to the surface of the membrane. But integral proteins will actually penetrate through the hydrophobic core, integrating into the membrane. They have a section known as a transmembrane section, a region known as a transmembrane region. And so we sometimes call these integral proteins, we sometimes call them transmembrane proteins, because it describes the fact that they actually run through the membrane. Integrins, which are a protein you've previously been exposed to, they're a special group of integral proteins because they can actually signal between the extracellular matrix and the cytoplasm between them. They represent one example of how particular proteins, particular transmembrane proteins, being placed in a specific membrane, in this case, that plasma membrane, gives a particular function. Here's just a brief example to show you what these proteins can look like. Here is water passing through one of these integral proteins, these transmembrane proteins. This is actually the same protein that you saw in that first wiggly video that I showed you. This is water passing through the protein known as aquaporin. Since water is hydrophilic, it does not associate with hydrophobic tails, and so it cannot pass through the core of that membrane without the assistance of something like a protein. So we've discussed a number of these different protein functions so far. We're going to discuss more of these, but I'd like you to take a moment before you begin our next video to look through in your textbook at the number of different functions, protein functions, in these membranes that have been discussed so far.